Lopinavir by Sarah Karbajakian, Matthew Koltes, Nelly Nazarian, Somi Uzuka, and Jennifer Worth. I put each of us on the structure as a functional group because for our project we all had our own jobs to do, and so we had to all function as a group in this at-home edition of Organic Chemistry. Sarah Karabajian, majoring in chemistry with a career interest in forensic pathology. Role is discuss 3D structure. Matthew Coltis, majoring in biochemistry. Interests are genius, indigent, scholar, philanthropist. Role is structural function as a drug. Nelly Nazarian, majoring in anthropology, bachelor's of science. Career interests are dermatology, and her role is function of a drug for COVID-19. Somi Uzuka, majoring in biology with a career interest in reconstructive plastic surgery. Role is discuss 2D structure. Jennifer Worth, majoring in pre-veterinary sciences with a career interest in veterinary medicine and a specialty in animal behavior. Roles are title, group intros, and background. Background on Lopinavir. Its molecular formula is C37H48N4O5. It is used to treat HIV and HPV as an enzyme inhibitor that stops it from maturing and prevents replication. It is commonly used in a 4-to-1 ratio with ritonavir. It was developed by Abbott Laboratories and was approved by the U.S. Food and Drug Administration on September 15, 2000. It is currently being produced under the name Kaletra by Abbott spin-off AbbVie, and considering it is not being produced by the Umbrella Corporation, it is safe to assume that there are no zombie side effects.
Hi everyone, so now we're gonna watch a short clip of Sarah discussing the structure of Lopinavir.
This is the 3D molecule that our group had, which was called Lopinavir. Now, if we rotate it, if we rotate it right here, we can look at all the different functional groups that we have, and all the dipole-dipole bonds, hydrogen bonding, and the shape. So if we look at the molecule closely, we can see our first functional group, which is the pyrimidine derivative. The next functional group, we have the amide right there. Next, if we zoom out, we can find the benzyl group. And if we zoom in, we can see that alcohol group we have over there. And then we have another amide group, benzyl group. And if we zoom in, we can see that ether group right there. And that one at the end is an arene. And then when we zoom in a bit, we can see a carbonyl group and another carbonyl group. And as we rotate a bit and zoom in, we see that carbonyl group, and as well as that ether group. The first type of hydrogen bonding we see is the NH when we zoom into that ring. Next, we have another NH as we rotate. And then that OH bond from that alcohol is another hydrogen bonding, as well as the NH group right there. The dipole that lopinavir has is right here with the carbonyl group that COH, as well as over here with the ether right here with the carbon, oxygen, carbon, and also right here with another carbonyl group, and then with another carbonyl group right here, and then also that NH group right here. As well as the CNH group here. Here's dipole there, dipole. The shape of lopinavir is, looks more bent. It does not look linear at all. And here is the 2D look of the molecule lopinavir. And this is how the 2D structure looks when it's zoomed in.
Hi everyone, my name is Nellie and Matthew and I will be talking about the functions of lopinavir as a drug. So, lopinavir is mainly used to treat patients with type 1 HIV and it's most commonly used with ritonavir. Ritonavir is a similar drug to lopinavir and you can see that the molecular formulas of the two are not too different. The reason why ritonavir is used together with lopinavir is because it increases the plasma half-life of lopinavir. It makes it a little bit more effective. Lopinavir's job is to act as a protease inhibitor. Protease is an enzyme that breaks down proteins. Those broken down proteins combine with HIV genetic material, which then form new HIV. The new or immature HIV will then mature and attack white blood cells responsible for our immunity. Case studies in 2012 have shown lopinavir's inhibitory activity against Middle East Respiratory Syndrome, or MERS-CoV, in vitro. Considering this, there are no compelling clinical studies to show lopinavir's ability to actually inhibit MERS in humans, but one particular study was conducted in Wuhan among 199 patients with the novel coronavirus to test the effectiveness of a combination of lopinavir and ritonavir. 99 were to receive a combination of lopinavir and ritonavir, and 100 were only to receive standard care. After a 2-week trial of administering the drug, the patients receiving lopinavir did not appear to be any different than those with only standard care. As a result, the trial concluded that lopinavir and ritonavir do not benefit patients with the novel coronavirus.
Good morning, everybody. This is Matthew. I wish you guys the best of luck in your presentations. I hope you enjoy the rest of ours, and I hope you guys are staying safe. So a quick rundown of the functional groups inside our compound. The blue oxygen with the blue hydrogen in the middle, that's going to be our alcohol group. There are 2 amide groups. Those are going to be the pink groups, which is the N bonded to the carbon, which is double bond to the oxygen. There's 2 of those. We have 3 arenes, which are going to be the red rings, one on top, the one on the left, and the one on the bottom leftish. There's an ether, which is going to be the oxygen that connects the top arene with the top amide. And finally, on the right, we have the pyrimidine ring, which is going to be 2 nitrogens that basically sandwich a carbonyl group. That's going to be the ring on the right side. And in this slide, we have 2 structures. The structure on the left is the lopinavir, that is the structure we're currently presenting. And the structure on the right is the ritonavir. These 2 are generally taken together. The lopinavir is normally taken exactly 4-to-1 ratio with the ritonavir. Both molecules resemble one another between the two pink amide groups. This is because lopinavir was derived from ritonavir. That's why they have similar structures. Where these structures differ is the rightmost double bonded oxygen. The carbonyl group for lopinavir makes up the pyrimidine ring, that's the orange carbonyl oxygen, and in ritonavir, the image on the right, it makes the ketone that combines the two nitrogens as well as the rest of the compound. That's the purple carbonyl oxygen in the right image. The next slide will show the identical structure that both compounds share. And for resonance effects, there are two kinds of resonance present in this particular structure. The resonance of the nitrogen-oxygen for both the amide groups and the pyrimidine groups contribute to a partial positive and partial negative charge on the compound, while the resonance for the normal arene groups, which would be the red rings, will contribute to a balanced charge. Both of these provide different contributions to the compound's interactions within the body. And yeah, between the two amide groups, you can see that they're exactly the same. The two nitrogens actually look very similar, but to be honest, there's actually one proton that switches between the two nitrogens depending on which structure you're looking at. Other than that, both compounds are actually identical. Resonance effects. So both positive charges and negative charges will contribute to dipole-dipole interactions, and the balanced resonance from the arene rings will contribute to pi-to-pi stacking. So the electronegative oxygens attract a majority of the electron density, creating a partial negative charge. This results in a partial positive charge around the nitrogens. These positive charges on the molecule are attracted to the negative charge on the oxygens within water, while the negative charges of the compound's oxygens are attracted to the positive charge on the hydrogens in water. This results in a dipole-dipole interaction, also known as hydrogen bonding. This increases water solubility for oral consumption and digestion. The three arene groups possess a different type of resonance interaction. There's no particular electron density from these resonance structures, although there is a slight attraction for the top ring towards the green ether oxygen right below it. Therefore, they contribute to pi-to-pi stacking, which increases hydrophobic, lipophilic, or just nonpolar solubility, as well as contributing to molecular recognition for binding. The two methyl groups on the uppermost arene provide restrictive rotation, since the two methyl groups tend to repel the ether oxygen during attempted rotation. This helps conform to the preferred target site, which ensures the binding at proper sites and reduces side effects. Side effects are produced when the compound binds with an unanticipated binding site, basically unintentionally. So lopinavir is normally administered in the 4-to-1 ratio with ritonavir, but why, you may ask? Well, when lopinavir is taken alone, bioavailability is limited to approximately 25%, so only about a quarter of it shows up in blood, while the rest is metabolized or broken down. This issue is not seen with ritonavir. This is because the ritonavir inhibits enzyme activity that breaks down the compound. When added to lopinavir, the ritonavir inhibited metabolization and plasma protein levels were steady at approximately 99%, significant levels for fighting infection. This would decrease the price, and 1,600 milligram doses would have to be taken instead of the normal 400 milligrams. This 4-to-1 ratio is currently marketed under the name Kaletra.
So lopinavir was FDA approved in 2000 and is a repurposed drug ready for trials. It is most commonly administered as a tablet under the name Kaletra.
So that concludes our presentation. But before we go, we would like to say a huge thank you to Professor Myers for giving us this great opportunity to connect and create this project that challenges us but still presents our individuality. Another thank you to my amazing and dedicated team of Nellie, Sarah, Jenny, and Matthew, who worked together to make this production come to life, which if I do say so myself, went pretty well. And finally, thank you to everyone watching right now. We really hope you enjoyed it and you all have an amazing rest of your day. Thank you. Bye.